Greetings, cannabis cultivators worldwide. Jordan River here with more GrowCast, and I'm cuckoo for Crobe'vember. Today we have Dr. Judith Fitzpatrick from the Microbiometer team on the line. This is a doozy. You may recognize Judith from the Instagram live streams that she does with the great Jeff Lowenfels. Well, she is here today to talk about microbes and soil health and the microbiometer. I really wanted to talk about the microbiometer. I think I'm going to do a video on it. I don't want to spend too much time on it here on the podcast and turn it into a commercial, but I do want to show you all how it works. So today we go into it a little bit, but stay tuned for a video on the microbiometer coming soon. Of course, code GrowCast now live. You'll hear more about it in this episode. And also just stay tuned for more Judith, because she's going to be back on talking more about soil science and testing and a lot more. Before we get into it, though, Photon Tech Lighting. That's right. You can find them at GrowCast Podcast /Photon. Code GrowCast gets you 10% off. I believe they're raising the discount to 10% off. We're working with a 5% discount code, but they're raising it. Shout out to Photon Tech. And use code GrowCast whenever you're hearing this. Cash in on that savings for the high-quality horticultural lighting from Photon Tech. This is a high-efficiency, high-output bar-style light. They've got the 465, which is perfect for a 4x4. They've got a 600, which is perfect for a 5x5. And if you want to get crazy, check out their CO2 1000-watt. Check out the Shane from MyGrow testing and review of this thing. He said it was too powerful. Take that for what that's worth. Basically, you got to run CO2 or you're going to overload it. I know you guys like power. Photon Tech has it. They also have efficiency. And now the new square models, the SQs. Check these out, everybody. This is perfect for like a 3x3 space, and I think they even have a 2x2. Very, very interesting. Smaller, but still powerful with that bar efficiency. Code GrowCast at Photon Tech. You can just Google them or find them quickly by going to growcastpodcast.com /Photon. Use code GrowCast. Send us a snapshot of you using the code, however you can get it to us. Also, be sure to check out our membership for more codes, more special discounts, and all sorts of fun surprises. Patreon.com /GrowCast. You got hundreds of hours of bonus content. You got the Discord you can get into. You've heard about membership. It's time you joined, everybody. We're having so much fun at patreon.com /GrowCast. I urge you to join up so you can catch the next GrowCast TV with Judith. I know you're going to love her. Thank you for listening and enjoy the show. Hello, podcast listeners. You are now listening to GrowCast. I am your host, Jordan River, and I want to thank you for tuning in again today. Before we get started, please share the show. Those Gromies who share the show are the best. They are indeed the most blessed in their garden. Be sure to join the green list for free. That's growcastpodcast.com /list, so you keep up to date on everything. And, of course, patreon.com /GrowCast for hundreds of hours of bonus content. Today we have GrowCast content, and oh my gosh, we have a first-time guest. I'm so excited to dive into soil testing, microbe health in soil, how soil biology affects cannabis. This is going to be quite a interesting deep dive today on GrowCast. And today's guest is none other than Dr. Judith Fitzpatrick. How you doing today, Judy?
Oh, very good, thank you.
I am so appreciative to have you on. Just for the listeners, to give a little background, Judy is a microbiologist. She is a kind of world-recognized leader in the development of on-site diagnostic testing, founding Cerex back in 1985. That's the company where she developed more than 15 medical diagnostic tests with a wide range of methods and reagents. Really, really interesting stuff here, Judy. I want to start just from the beginning and eventually work our way into how you came into cannabis, of course, coming onto the team with the Microbiometer, who we're partnered with, working with the great Jeff Lowenfels, who we know and love. But to back it up before that, I mean, you were part of this microbiology thing in this on-site testing way before it was cool, way before, cannabis came along and all that, right?
Right. That's been one of my big areas of interest, and I have quite a few patents in that area.
Oh my gosh, that's so cool. Well, I mean, what was that like 20, 30 years ago, and now how it's developed over the decades? If you could just kind of give us a background on how you came into the field and how it's evolved over time.
Well, it's become, we were in medical diagnostics, so our tests had to pass the FDA. And the first test I worked on was at Becton Dickinson. It was a pregnancy test.
Oh, I had no idea. That's fascinating.
Yeah. So, then I founded my own company and I had the first commercial assay for detecting whether people smoke using serum or urine.
Whoa.
Yeah. So that was the insurance companies used that. And we went on to develop the first quantitative strip. And it's still for sale by CBS. It tells you how much you smoke. I didn't develop it for how much you smoke because I don't think person at home would take a test to know how much they smoke. They know how much they
Right.
smoke. But I was very interested in being able to quantitatively figure out, dangerous drugs like those are taken for epilepsy. And
Right.
in children, they have to be monitored very closely because children's metabolism, varies so much from day to day that the levels can go up
Right.
too high or down too low. So that's kind of where I started going with that. And I worked on one developed one for Bristol Myers too that was for osteoporosis.
This is just so fascinating. I knew that you were into the biological side, but I didn't realize that it all started in kind of a medical setting, like you said, for various insurance tests. That's really, really cool. How
Yeah.
did what bridged that gap?
Well, what happened was I sold my company in 2000 to take early retirement.
Sweet.
And I always wanted to teach. I had done some teaching when I was younger. And I went to the local college and they said, we'd like to get some National Science Foundation grants. And they were meeting with me about that. And then one of the founders, James Satillo, who was a leader in organic and maintenance of public places. Like he maintained Harvard lawns and all of New York City's outer spaces and everything.
That's so
He
cool.
came to the college and he gave a talk on that. And it waked my eyes to the fact that the earth had lost 50% of its soil. And without microbes, you had no texture to your soil. It was just eroding,
Right.
that we weren't getting the nutrients and the taste in food that we needed. And so I said to him, and he said, this is what we need, a test for microbes, because that's the one thing that really tells you what's going on. And it needs to be cheap and it needs to be easy to do. So it took a long time for us to develop it in the strip format. And from the beginning, I was joined by my partner, Brady Trexler. And he's the one who does the phone app and adapted it to the phone. And we are the only test I know of where you take a picture and it gives you a quantitative answer from the picture itself.
So super fascinating.
Yeah, he's brilliant. Brady is.
I'm just so fascinated by this evolution and what kind of links this academic, long-standing, and so fascinating field of like traditional regenerative agriculture and what it was pre-cannabis, and now this wave, this green rush wave of cash crops to study.
Well, I think that the really interesting thing for us, we didn't start out thinking about cannabis.
Right.
Okay. But, there hadn't been any new tests in agriculture in 100 years. Nitrogen, phosphorus, potassium, they were all done the same way they were done 100 years ago.
That's a really good point.
And people knew that they weren't getting all the information they actually needed from those. They needed other indicators. And one of the best indicators was microbes.
Of course.
So he said he wanted that test. And I had an NSF grant, National Science Foundation grant, for training students in quality assurance because I ran an FDA facility. And so when we finally got this test developed in its very simple form that it is now, it was so revolutionary that the people in agriculture were saying, this is snake oil. Nobody can do this. That's really what they said.
That is too funny.
People I work with now.
And you're referring to the microbiometer itself, this device that again, if you're on the green list, you've heard of this. If you're in membership, let's just throw it out there. Microbiometer.com, just like it sounds, spelled just like it sounds. Code GrowCast will get you savings. at-home microbe test kit. Distinguishes between fungal and bacterial. We're going to get into all of this more deeply, and gives you a quantitative level of living biology in your soil, in your compost teas, and more. It's affordable, and it's even more affordable with code GrowCast. I think it's 15% off. So go and try it out. Again, this is kind of, it's a product that really interested me. Obviously, if you've been following the show, you can understand why. Several team members and friends of the show are already on this. Riza Rich is the one who introduced this to me. And, Dr. Judith Fitzpatrick is part of the board, but also you may recognize another board member, former guest of this show, Jeff Lowenfels. So they've been around for a while, and I'm just really, really happy to be partnered up with them. And yeah, I want to go deeper into how microbiological testing works, what it can and can't do, and what the practical applications are for home cannabis growers and home gardeners of all kinds. So there's kind of a 30,000-foot view for the listener. Where do you want to start with this, Judith? What I would really like to focus in on is this idea of home testing, again, what it can do, telling you levels of broad kind of ranges, but then also what it can't do, distinguishing specific species and what that takes. I would be very interested to learn more about that process as much as you want to share here.
Okay, so broadly, we can tell you we correlate with the $550 laboratory test called chloroform fumigation assay, which tells you microbial biomass. And we're $10 as opposed to they're $550, and they don't tell you the fungal to bacterial ratio. Okay, so we've got even a bonus. And we correlate with the CFE test 96%.
Oh, wow.
So that's, basically that's amazing. And we work with Dr. Forbes Walker at University of Tennessee, and they worked with on this, and he's using the test. And so they're using it at Cornell. They're using it at City University. They're using it at Stony Brook and other universities now, because it lets you follow what you're measuring. Now, basically, to a large extent, what the microbe is telling you that you have a balanced and adequate amount of nutrition to feed the plant. If you were not putting down chemicals like nitrogen, phosphate, and potassium, etc., all of those nutrients would come to the plant via the microbes.
Right.
And the plant nourishes this population by putting out 30 to even 50% of exudates through its roots. And at different times, it puts out different exudates to bring in different microbes.
Mm-hmm.
If you feed your plant heavily, like people do in conventional agriculture with these minerals, I mean, you can use a little bit of them.
Sure.
If you feed it heavily like that, the plant says, I don't need to feed these microbes.
Right.
And this is disastrous because the immune system of the plant is the microbes.
Mm-hmm.
Without the microbes, the plant does not develop an adequate immune system. So in a big study published in Nature, covering something like 800 farms, organic farms in Europe, what they showed that they used 97% less pesticides.
Wow.
Even organic pesticides.
Wow. Because of the immune systems.
Right. And then the other interesting thing about that is these plants do get visited by pathogens, and the pathogens stimulate them. And the stimulation of those plants, including your cannabis plant, causes your plant to make 2,000 different anti-inflammatory, etc. type compounds.
Wow.
That basically increase the nutrients of your food.
That is fascinating.
Or your cannabis. If you go to when I go to the show, the smells of these cannabis are fabulous. And the reason they smell different and that they have different flavors is because that soil that they have is hosting a different microbial population.
Mm-hmm.
Just as any land is to some extent, any tropical or Arctic, is going to like have a certain group of people who have a certain group of skills.
Yep.
So the same thing is happening in that. And so it tells you that you have everything there that you need for the plant if you have a good microbial biomass.
Sure.
Oddly, when we see sick plants, we see very low microbes.
Wow.
I have never seen high microbes by microbiometer in a sick soil.
And the plant looking sick, right?
When the plant is sick,
Right.
I mean, I've gone out. There's a guy next door to us who keeps planting his $500 tree. He's planted one. The nursery's come back 3 times and put it in. And on either side, there's nice trees. They're reading 400 or 500 microbes, their count. This hole, it's like 50.
Whoa.
I mean, the guy should be testing it and say, there's something really wrong. I've got to figure out what it is. Now, our test will not tell you what it is.
Sure.
Okay. There could be a toxin there. Somebody could have buried something very toxic there a long time ago. So
Jeez.
It's likely that it has the right amount of chemicals in it because I'm sure the chemicals diffuse throughout the soil. It's very close to the soil. It reads very well. But I can't tell you what's wrong with it.
Sure.
But here's the thing. When you put down any kind of amendment, it has to be really special for your soil and your crop and your cover. It's like anybody who goes to the doctor. You can't imagine that the doctor has one pill that's going to cure everyone.
That's how we kind of got into our American health crisis right now, is what it seems like. But I digress. Go on. Yes, I agree.
Yeah. So, I mean, what you need to say is this amendment actually what my soil needs? Maybe it's giving the nitrogen that your plant needs, but maybe it's giving too much sulfur.
Mm.
And here's the other thing. Some of the organic farmers I work with, they'll tell me, like, my microbes look good. My chemistries I get from the lab are terrible. But I've had my food tested. It has plenty of all these things in it. Where are they coming from? They're coming from the microbes. A lot of it is not like chemically available to chemical tests, like phosphate. There's too much phosphate in most of the soils in the world.
Right.
But it's only available when the fungi make it available.
Mm-hmm. I see.
The fungi actually dissolve it from rock and from, mineral material.
Right. So that and this makes sense to me when you talk about testing the levels to get a picture of the soil. But I'm also interested by what you just said a minute ago, which is it's also a broader picture. Like you said, you've never seen, the microbe count be high and the plant just be sick. It kind of shows a broader scope of what's going on. I do appreciate that. I think for the listener, though, maybe again, just for a 30,000 foot view, this microbiometer as a home test, it's just kind of a home slurry test. You mix up the soil sample in a little tube and you kind of homogenize it in some liquid. You let it sit.
So you put 3 drops on a little test card we give you.
Right. That's to get a pigment, right? To get a color.
You take a picture.
Uh-huh.
When you do a good soil, you can't see through it. There's so many microbes. And then when you do the microscope, the whole top tube is all microbes. It blows everybody's mind when you first see how many microbes are there. We take the tiniest amount of soil. It's like maybe 1/8 of a teaspoon, and we use 10 mils of water of an extract fluid, and we whisk, which separates them because they're stuck to the soil. In a lot of the other tests, they're looking for something very small, like the biological testing, and then they multiply by 1,000, assuming, or 10,000, or 850, whatever number they come up with. But we are able to microscopically count everything that we have separated from the soil, and these soil microbes are colored.
Wow!
And we detect the color. And we capture them on a membrane. The other fluid goes through. We take a picture, and depending on how dark it is, we can measure how dark it is. And based on that, we can get a curve just like you do in a laboratory test, and we can tell you how many micrograms per gram of microbial carbon there is.
Gee. That's insane. But doesn't it give you a readout to your phone too, like a historical readout, or do I have that
yes,
wrong?
when you take
Okay.
the picture, it takes a picture, and then it just immediately tells you, the fungal to bacterial ratio.
That's so cool.
Yeah. So
That's
actually,
so cool.
the camera takes like 20 pictures real fast. Like within 1 or 2 seconds, it's taken 10 pictures, and then it does an average of the 10 pictures.
Man, that is fascinating. And again, distinguishing, like you said, the ratio between fungal life and bacteria. Very, very impressive. What type of practical applications have you seen people using this for? My understanding is that you can use it to test your compost teas. Obviously, you can test the levels in your home garden, your home grow soil. What do you see like cannabis growers using this for practically?
Well, if a person decides to, try to see which amendment or which fertilizer would work best in their soil, okay, then they can try this treatment like I do. I've been recently using fish shit.
Oh, hell yeah. Shout out to fish shit.
Yes. And so I put that on, and it's a lot of nutrition, and I can see within a week or two that I've increased my microbes.
Wow.
Like a lot of plants, as they go into seed or go, as corn starts to ripen, the microbes and and then the amount of nitrogen in the soil go down. So nitrogen actually correlates with microbial biomass like 90%.
Oh, that's
And
fascinating.
a lot of people have seen that, and so does phosphate, and so does depending on the methods. If you use really sophisticated methods, you can get generally speaking, there's a good correlation between all of those things.
Right.
There's an excellent correlation. So we know that especially fungal metabolites are present in the cannabis plant, and they're present in the flowers,
Mm-hmm.
and they're present in the elixir. And to a large extent, that's what gives flavors, etc., to these things. And
That's what
also,
gives
we
the
know,
flavor? Oh, no, you're chopping in and out a little bit, Judy.
yeah,
But you're saying that the presence of fungus is directly affecting the flavor of the flower. That's what you're saying?
yeah. So the plant allows microbes to invade it all the time, just like we have microbes within our body.
Mm-hmm.
And if it gets an infection, you don't necessarily get sick. You get infected all the time and you don't get sick.
Right.
So when that happens, there are microbes that the plant knows it needs. So it basically sends out chemicals that bring basically those chemicals are foods that attract the bacteria that make those compounds, and those bacteria or their metabolites enter the plant and disease and things like that. And, it's a big problem, interestingly, in hemp, because if a hemp plant gets stressed, it makes too much THC.
Right. And then it fails testing.
So the amount of stress that a plant that is grown organically or, sustainably, like with not a lot of chemicals, does have this strength, and it does produce more cannabis.
And this is super fascinating. I do want to kind of double back a little bit on what you said. I think is a very interesting point. Talking about how well a product might take to your garden. This is something I've heard time and time again, is that different species of microbes are going to thrive and dominate different environments. I hear this about mycorrhizal fungi, for instance. And people say, why are there multiple species of, glomus or whatever in this product? And usually the answer is because one is probably going to do better in your situation and become dominant and thrive. A, do you agree with that? And then B, this is a way to test that. This is a way to directly test if the compost tea takes better than the fish shit or whatever, just for example, right? Is that an accurate statement?
It's very accurate. I mean, that's exactly what's going on. you've hit the nail on the head.
And it varies from person to person. It literally might be way more effective in one garden than another because that's just how agriculture works.
Yeah, I went to University of Mississippi one time and tested the soils, and what we found was very interesting. Like the amount of microbes in the soil in a sustainable or organic system correlates with the health and yield of the plants.
Wow.
It doesn't correlate in a chemical, a mineral fertilizer regime. Okay, because you can push the soil, but then you're destroying your soil. You're going to lose all
right.
Over
There's
time.
a point of diminishing return there. Yeah.
Right.
Man, that is very fascinating.
Yeah.
I'm thinking of the implications. Again, like testing different products for your garden.
Well, what they found at the University of Mississippi was they had 3 different fertilizers, 3 different crops, and 3 different cover crops.
Mm-hmm.
And the fertilizer that was best depended as heavily on the match of the cover crop and the crop.
Wow.
The thing that really fascinates me is that we're dealing with a community.
Mm.
These microbes, as I love to say, have survived for 4 billion years.
Right.
Okay? No mammalian species has ever lasted more than 100 million.
Mm-hmm.
So they are incredibly adaptive. And they pick up genes from one another all the time. So they mutate. And they can mutate to respond. That's why they've survived.
Right.
And they defend their territory.
Mm-hmm.
So if they don't like what you just put down, they're going to kill it.
That's what I heard. That's why we have these plastic-eating microbes we're discovering now. We're putting down so much plastic, the Earth is going to fight back eventually. Is that kind of what's going on?
That's exactly right. They can mutate for good and they can mutate for bad.
I love it. I fucking love it. That's incredible.
Yeah, they just are incredibly resourceful like this. And they have very small amount of DNA. Okay, so we have 10 to 100 times more DNA than a microbe. We have to eat certain foods because we don't make them.
Mm-hmm.
Think how dependent that little microbe is.
Mm-hmm.
He depends on other microbes for some of his vitamins, for this, for that. So you're not just walking in and saying, I can put this microbe down. I mean, it's like opening a whole bunch of Xerox stores in one town.
Wow.
You have to match what the needs of that community are.
That's fascinating. Can I, kind of on that subject, I'd like to ask you a kind of home grower specific question, and maybe there's no correct answer, but I'd like to get your thoughts anyways. I know it'll be good. There are 2 kind of different categories, I would say, of microbial products in the cannabis world right now. And this is a broad statement, but I'm just going to kind of frame it up for you. Some are more specified, where it's like a couple species of very specific bacteria, photosynthetic bacteria and, some sort of phosphorus solubilizing bacteria and stuff like that. Whereas others, like the aforementioned fish shit, are kind of more scattershot, broad compost tea products, things that have a lot of that BioCat product, lots of different diversity. Do you think there's an optimal way to go? More specialized microbes? It almost sounds like you're saying it's all about the diversity. I don't want to put words in your mouth, but that's kind of what I'm hearing.
Well, my philosophy is that the main thing you're putting down with many of these things, I mean, there are some cases where the microbes, are able to colonize under some conditions. But I think for the most part, I mean, we've taken compost and boiled it, and we still get the same effect as if it's not boiled.
Hmm? Yeah,
because, a microbe divides as rapidly as every 15 minutes. Like you can get 100-fold increase in microbes overnight. Just put a little sugar in a vial.
Right. Yeah,
So
good point.
I think that for a lot of people, the answer is feed your microbes. So that you get a healthy population and let that population modulate itself, which it's going to have to do over time. And it is going to take some time. I mean, people come to us and they say, here's a plot that is just conventional, and I just put down some nice organic stuff on this plot next door, and it's the same number of microbes. Well, you don't cure a land or get a healthy population going in 2 weeks, usually.
Right.
You have to build it, and some of those species, the diversity that we're talking about, it has to be a diversity that's adapted to your crops and to your soil type and your climate.
That makes perfect sense. Fascinating stuff.
Yeah, and I've talked to some of these people who some of the companies we work with are making, looking at microbial diversity, and we know that microbial diversity is associated with better health of the soil. But I've asked them, and they concur that I'm right. The diversity has been there all along. It's just that when it's very low, you have a very small number of any one species, and you can't pick it up by the test.
I see. Yeah. And you know what? I think that you're also primarily talking about some of these agricultural setups here. A lot of home growers, they're not outdoors. They're in it's a little bit different when you're working in like a modified growing medium inside versus the situations like you're talking about. We'll be right back with Dr. Judith Fitzpatrick. But first, Plant Revolution. PlantRevolution.com will show you more information about their products. Your local hydro store carries them, I'm sure. It's time that you try them if you haven't. That's right, they make the king crab. You see me wearing the king crab hat. Well, I don't just like the hat. I love the product. It's beneficial bacteria, and it is extremely clean. That's why I recommend king crab to everybody. It doesn't matter if you're in DWC or living soil, it is applicable. I'd be worried about adding some of the more natural, stanky microbe products to, for instance, a sterile DWC system. You don't got to worry about that with the king crab. Same goes for the orca. The orca is their liquid mycorrhiza product. Now, you see a lot of myco in granular form, but if you need to add it to your, say, DWC system, or let's say you may have hammered your soil with an antifungal agent and you need to re-inoculate it without transplanting or tilling or anything like that, add the liquid orca mycorrhiza. You will see results very, very quickly and very, very noticeably. They've also got the MycoChum to keep all those lovely microbes thriving and eating, and so much more at PlantRevolution.com, everybody. Go and check it out. They're great friends of the show, and stay tuned, as always, for mad giveaways in membership, at classes, and that's all thanks to Plant Revolution. All right, let's get back to it with Judith. And we are back, everybody. Me and Dr. Judith were just talking off air. You said something really interesting about the difference between farmers, traditional farmers, and their willingness to try and honestly risk new practices versus cannabis growers who will try anything, right? You want to talk about that?
Yeah, so we really, owe our start to the cannabis industry, which was, really picked up on us very early because they're young. They love cell phones. We were thinking we would go into traditional agriculture, but the average farmer is over 60.
Sure.
He's not that comfortable with a phone.
Yep.
And traditionally, he sent everything out to the lab, and the lab and his testing is paid for by the fertilizer company.
Right.
And then based on the test results, the fertilizer company comes back with recommendations for the fertilizer.
Yeah. So it's all self-contained, and this is kind of a decentralization of the power, but also takes a degree of education.
He's never made his own decisions.
Yeah.
And the cannabis grower he was looking for information, but he knew he was on his own.
Yep. Yeah, exactly. We were already on our own to begin with. That is super fascinating. And again, it is a pleasure. I mean, this episode is more than just a pitch for the microbiometer. I'm so stoked to be partnered up with you guys. But you are obviously a wealth of knowledge when it comes to microbiology and soil health. And anytime you see a board of directors or whatever you want to call it, like you guys, it's just it is really exciting. We're on this note though, so I want to double back to something I said before the break, which is a lot of these cannabis growers, these young cannabis growers, like you said, they're growing in a tent inside. Even if they're organic, they'll buy like what's considered a super soil, like a rich organic soil loaded with nutrients.
Right.
And in reality, that's just like a peat moss base usually. Usually it's got perlite or maybe rice hulls for aeration. And then it's just got its, mineral, nutritional inputs, whatever it may be. They all have their own recipe. But you're not working in a native soil usually. Usually you're working in a container, like a 3 to 10 gallon pot or grow bag or fabric pot. And usually these people are hammering that small space with all sorts of products, like trichoderma one day, and then they're adding liquid mycorrhiza, and they'll throw in some fish shit and some king crab, and they're just pouring it on. It's not the same as the outdoor scenario. It's not the same as native living soil, part of that community. Like you said, it's almost like a space colony to that community. You know what I mean? Like the first Mars space colony is to us as the microbe life in the tent is to the microbe life out in the earth. Is that a fair analogy?
Well, I think it is, but quite a few people we worked with in the field actually recycle their soil.
Mm-hmm.
And they do that because what they've found is like the mycorrhiza that have adapted very well to their plant, it sporulates. So now that soil has all those spores in it. And it has a microbial population that's adapted to the strain of cannabis that's being grown, et cetera. So the danger of that, of course, is that if there's any disease going around, it would stay in it. But, they are on the watch for that. But I think that's not a bad idea. Obviously, the soil has to be replenished and stuff like that. But it's not a bad idea to, at least use some of these soils for reseeding so that you're using, even now people say the best mycorrhiza that you have are basically the mycorrhiza that are inherent in your soil.
Wow.
So when they're telling people to encourage mycorrhiza, they're saying add some of your soil to your compost.
Right.
So that you're bringing in your native species.
Right. That does make sense. And when you mentioned the reusing soil, that's another thing that a lot of people don't do. And you're right. You took all this time to build up that established colony of whatever one ended up being dominant, even if you were using trichoderma on top of your mycorrhiza or whatever became dominant is in that soil. So maybe, like you said, replenish it, re-amend it, and use it again, or use it as seed starter. Great question. Let me ask you this. Could you end up using a portion of it to reestablish and repopulate, or is it better to go whole cloth?
Oh, I think you definitely could.
Yeah?
I think that's, I'm not an agronomist or anything, but that's what people do with compost. They'll put in some of the soil that's naturally found around their area and inoculate the compost so that it contains the native microbiome.
So just a little shot in there and mix it up and reestablish some of that life.
Yeah, those guys have a good community. They can support one another's. They have a thing called plate count. You dilute your sample and you put some on a plate of agar, which is food for them. And then you look to see how many microbes come up.
Mm-hmm.
And you can count them. That's one of the ways people figure out, how many microbes are in the soil. But you can only really culture about 1% of them because they're not adapted to agar plates. They're adapted to soil. But I did it one time and the school was closed. The college where I was teaching was closed. So because of a storm. So I couldn't get in for a week.
Oh.
And I had seen them after 2 days, which is when one usually looks at a plate. And there were, maybe 8 colonies. And I came back 10 days later, and there were like 23.
Wow.
And they were all different colors. So it was not the same.
They were adapting.
It wasn't like one of these doing. I let it go for 6 months, and continually new strange looking colonies emerged.
It's so fascinating. It's the adaptation that you were talking about.
A lot of these microbes are dormant. And when the population around them begins to supply the foods that they need, they become active.
So wild.
And it just goes on and on. I just kind of wish I'd done a year, but it was pretty crowded at the end of 6 months.
It's absolutely incredible. Honestly, from my perspective, it's all kind of Greek to me, and I hope to get people on like you on the show, and then kind of disseminate this information to the cannabis growers. But I do look up to people like you, and I'm super fond of like hearing those stories. We'll have to have you back on if you're interested, and do more story time. Like I just absolutely love it.
Okay, good. And maybe we could share some of the stories of some of our some of the people who are doing research in different parts of the world.
Sure.
On this type of work and everything.
That sounds incredible. Before we wrap up this episode, I do have one question, and I don't know if this will be a flop. I think it's going to be a hit. You see, I do another show called the Coffee, Health, and Science Podcast, and we talk about coffee a lot. We talk about health as well. And the subject of the microbiome just ends up dominating that podcast because so much of health and wellness is being rooted back to the human microbiome. We talk about the gut microbiome. We talk about, the vast majority of disease originating from the gut. We talk about the microbiome on our skin, on our eyes. We talk about the over-sterilization of our food. We talk about the over-sterilization of our bodies, what that does to the microbiome and what that does to our health in the long term. What we've really come to find on this podcast, what this kind of chain that we're discovering, is that the human microbiome really starts with the soil microbiome.
Yeah.
And that when we take a look at these synthetic farming practices that harm microbiology, we take a look at them and we're like, oh, we focus on things like nutrition. What's the nutrient value? But there's a bigger picture here, a picture that we barely understand. And all we know is that the microbiome in our body is extremely important to maintain for our health, and that it comes from the earth. And that if you disrupt that chain, we're in big trouble. That's virtually, I mean, listen, I've talked to leading experts, and I'm here as a layperson. That's virtually all we know. We don't know much. Do you agree with that? Have you seen this link between the rhizosphere and the human microbiome?
Yeah, and I think that's fascinating. And what made it, rather easy for me, see, developing diagnostic tests is immunology, because my tests were all immunological.
That's crazy.
So, I'm basically an immunologist, a chemical immunologist, basically. So you see the same thing, like when you take an antibiotic, you're killing not just the bug you want to kill, you're killing so many other different bugs in your body. And it makes you so susceptible to disease. And it so unbalances your whole system. Don't even get me started. I knew this was a good question. I'm going to have to get you on the other show or something. This is great. Oh, I'd love to talk. I mean, one of the things people don't realize is you always have strep in your throat. Right. It just overgrows under certain conditions. Fuck, so I love it. So when they say you have a strep throat, it's because you have too much of it. And your body usually likes to keep a little bit of that because it says, like Lincoln said, keep your friends close, but keep your enemies closer. You get to know them real well when you've had strep for that long. But that's also just like the soil balance. It's the same exact thing. Exactly the same thing. Oh my God. Botrytis is always there. It's always there. Yeah. All of these pathogens and all of these things are always there. And the microbes do a really good job of balancing the whole thing. Wow. Events can happen that really stress them. But, you take AMF fungi. Okay. If you take AMF fungi and you look at the root of a plant during a drought, a plant that doesn't have AMF, the root is narrow and small. Yeah. Mm-hmm. The root is big in a plant that has AMF. It swells up because the fungi tell the plant to put sugars in there. and it makes the osmotic pressure get bigger. And that allows the plant to absorb a lot more water.
Whoa.
So it's this communication between the fungus and the plant. And why is the fungus doing it? The fungus depends on the plant for its nutrition.
Just incredible.
And the plant depends on the fungus for phosphorus and nitrogen.
That symbiotic relationship. And that's something I've observed again in coffee before, the organic farms surviving the drought down in Colombia. I mean, a story for another time. Unfortunately, we do have a hard out here.
Yeah.
My gosh, Dr. Judith Fitzpatrick, give it up, folks. The audience is, you can't hear them, but the podcast audience is clapping. What a wonderful first appearance this was, and I would love to have you back on again soon.
Okay, it was fun talking to you, Jordan.
All right, we'll do a little. I'll do a little wrap up here, and then we'll talk off air after. But for the listeners, of course, microbiometer.com, microbiometer.com. Go ahead and pick up your home microbial testing kit. Use code GrowCast for 15% off. Stay tuned for more episodes with Dr. Judith Fitzpatrick. We're gonna have the great Jeff Lowenfels back on the show. Holy crow! So stay tuned, everybody. This is Jordan River and Dr. Judith Fitzpatrick signing off, saying to you out there, be safe and grow smarter. That's our show for today, everybody. Thank you for tuning in again. Before we wrap it up, AC Infinity, one of my favorite partners of ours, ACinfinity.com. Code GrowCast15 for 15% off. The best fans in the game, the sexiest tents you'll ever find, fabric pots, ratchet hangers, scissors, basically everything you need to grow, almost everything you need to grow, over at ACinfinity.com. They've now got lights up there, the ion grid. These things are freaking beautiful. They even have an onboard display. I'm hoping to get one of these. I think it's on the way. At least as this episode is dropping. ACinfinity.com. Get what you need to get started growing. Trustworthy, American-designed products, always innovating. ACinfinity.com, code GrowCast15. Thank you all so much. Be sure, like I said, to check out membership at patreon.com/GrowCast. So much fun stuff I'm cooking up in there, you guys. Get on the green list. That's free. Go find that at growcastpodcast.com/list. We're doing giveaways there too, plus resources and quick grow tips. I am having so much fun producing all this content for you folks. I hope you're learning an incredible amount from the show, or at least a little bit. And it's truly an honor to serve all of you who are serving your plants within your own garden. So stay tuned. We've got new guests. We've got old guests. So much planned for this month that you're going to love. I appreciate each and every one of you subscribers. We'll see you next time, everybody. Bye-bye.